37. Canal systems. Ascenoid flagellated spongial, simplest body form, small and tube-shaped. Water enters a large open cavity, usually in the middle of the spongial cell. Lines with choanocytes. Choanocytes, flagella, pull water through. All Cacaria are ascenoids. Ciskanoids. Flagellated canals resemble ascenoids but larger with a thicker body wall. Wall contains choanthocytes, lined radial canals that empty into spongial cells. Water enters radial canal through tiny openings called procytes. Spongial cell is lined with epithelial cells rather than choanthocytes. Food is digested by choanthocytes. Flagella draws water through internal pores called apophylles into the spongial cells and the osculum. Developmental evidence is being derived from ascenoid ancestors. Leuconoids. Flagellated chambers. Most complex and are large with many oscula. Clusters of flagellated chambers are filled from incurrent canals and discharge to excurrent canals. Most sponges are leuconoids. The leuconoid system evolved independently many times in the sponges. System increases flagellated surface compared to volume. More color cells can meet food demands. Large sponge filters 1,500 liters of water per day. 38. Modes of reproduction. Gemmules, internal buds. Regeneration, when a portion of the animal is wounded and can heal over itself. Sexual reproduction. One sponge releases sperm, which enters the pores of another sponge. Choanthocytes phagocytize the sperm. They transfer to carrier cells. Transport sperm through mesohall to oocytes. Monoecious, having both female and male cells in one individual. Sperm sometimes arise from formed choanthocytes. Oviparous, formed on the outside of the body, larvae have to settle on the solid surface. Viviparous. After fertilization, the zygote is inside the parent and is nurtured there. 39. Class Cacaria. Spicules of calcium carbonate. Spicules are straight or have 3 or 4 rays. Most are small but tubular or ray-shaped. Ascenoids, ciskanoid, and leuconoids are body forms. Examples are Eupecta, Viscinolia, Porterium, Calispongia, Platarina cariensis is common on Caribbean reefs and caves and under ledges. Sesquentillatum, one large terminal osculum bridged with a crown of spicules. Class Hexamelida, glass sponge with 6 rayed spicules of Sicilia. Silica, nearly all develop seed form, or deep sea. Most are radial symmetry. Stalks of root spicules attach themselves to substrate. Spicule system forms a network. Tubular reticulum is the largest continuous synthetic tissue known in metazoa. Choanoblasts are associated with flagellated chambers. Choanoblasts are located in the primary. Each has one or more extended two-colored bodies. Lack of pinociderum or gelatinous mesohell. Myocytes are absent. Chambers appear to correspond to both ciskanoid and leuconoid types. Deep water habitat with large and easy flow of water. Eupecta aspergillum, glass sponge, Venus flower baskets, brittle, and young shrimp can crawl into them when they're very young and get trapped. Class Demospongiae contains 95% of all sponges species. Spicules are siliceous but not 6-rayed. Leuconoid body forms. All marine except spongulate, the freshwater sponge. Fresh water is widely distributed in well-oxygenated ponds and springs. They flourish in summer and die when it gets cold in late autumn. leave behind gemmules. Reproductive sexually, but existing genotypes may also annually form gemmules. Marine demosponge, highly varied in color and shape. Bath sponges, Laxacilius spicules, have sponge skeleton, are found on Caribbean coral reefs. Spongilla, fresh water, crust-like branched or clumped, texture fragile and soft, color reddish or green, irregular scate or barely visible oscula, surface uneven and roughened by spicules, live in harsh environments, very cold. 40 is included in 39. 41. Phylum Cnidaria, general feats. Tentacles surround the mouth or oral region. Sense organism for balance, zoocytes. Photosynthetics is ocelli. Nerve net, symmetrical and asymmetrical synapses. Diffuse conductation. 2 nerve rings in hydrozoa and medusa. Muscular contradictions via epithermomuscular cells. First animal to show belong to the Eumetazoa clade, but all but sponges. The oldest group in the clade is the cnidarians. Fossils date back over 700 million years ago. Over 10,000 species. All are aquatic and mostly marine. Incomplete gut, gastrovascular cavity, or GVC. Extracellular digestion in GVC. GVC. 42. Radial symmetry or biradial. It could be divided unlimitedly. Polyp. Hydroid form, adapted to sedentary lifestyle. Tubular body with mouth directed upward and surrounded by tentacles. Mouth leads into blind GVC. A boral. Surfaces away from or opposite the mouth, and attaches to substratum by petal disc, a thin plate of tissue by means of which the animal adheres to the substratum. Reproduce sexually by budding, fission, or petal laceration. In colonial forms, polyps may be specialized for feeding, reproduction, or defense. In class Hydrozoa, feeding polyps, hydrants, distinguished from reproductive polyps, Gonangia, by absence of tentacles in Gonangia. Medusa, bell or umbrella-shaped, usually free-swimming, mouth directed downwards. Tentacles may extend down from rim of umbrella. Medusae, equipped with steocytes and ocelli. Integration of sensory information into motor response. Function of a nerve ring located at the base of the bell. Cnidarian lifestyle. Typically zygote develops into motile planula larvae. Planula larvae settles in metamorphosis into polyp. Produces other polyps asexually. Polyps eventually produce a free-swimming medusa by asexual reproduction, budding, or stropulation. Medusae, dioecious, reproduce sexually. True jellyfish class, Saphizoa. Medusa enlarged and conspicuous. Polyps typically very small. Most colonial hydroids. Features of polyp stage and pelagic medusa stage. By some hydroids, Ulysia, form floating colonies. In hydra, only stage is a small freshwater polyp. 43. Diagram and describe nematocysts. Tiny capsules containing a coiled filament. Filament may be tiny barbed or spined, except in Anthozoa. Cnidocytes equipped with trigger like Cnidocyl, modified cilium. Tactile stimulation causes nematocysts to discharge. After cnidae discharge, cnidocytes absorb and another develops.